Candida albicans is a commensal fungus typically found in gut, mouth, and skin of most healthy individuals. However, it also can become opportunistic fungal pathogen that can cause infections on the surface, like mucosal membrane, and also can cause life-threatening condition. This fungus has the ability to transition between yeast and hyphal form. This transition is triggered by environmental factors and signaling pathway. There are three characteristics of fungus. First is yeast, which has a round or oval shape and a unicellular morphology. Second is hyphae, characterized by its tubular structure and multicellular morphology. A unique feature of hyphae is that it has long, continuous structures without constrictions between cells, resembling a tail. Lastly, pseudohyphae have a chain-like structure and are multicellular. Pseudohyphae are elongated and formed by a series of yeast cells that remain attached after division. Candida species appear under a microscope in the yeast form, with long true hyphae or pseudohyphae, and also exhibit a characteristic shish kebab appearance. In order for hyphal morphogenesis to occur, there are several environmental factors that act as the trigger. The factors include host characteristics, which are elevated body temperature and carbon dioxide level, low glucose concentration, and alkaline pH, and also oxygen level of the fungi. Not only that, the presence of N-acetylglucosamine, amino acids sensing and quorum sensing can also lead to hyphal morphogenesis. The first pathway that plays a critical role in hyphal initiation is cyclic adenosine monophosphate, CAMP, protein kinase A, pKa, pathway. This pathway controls the C. albicans pathogenicity, morphological changes, sexual reproduction, stress reactions, and sensing and acquiring nutrients. CAMP-pKa pathway induces transcription via FG1 or Flo8 to exhibit the functions mentioned before. Next is the SIC-MAPK, mitogen-activated protein kinase, pathway, which is activated by embedding matrix environments, damage to the cellular wall, and insufficient nitrogen sources. This pathway involves the activation of transcription factor CPH1, which is a positive regulator of filamentation of C. albicans. Let's go through the first step, which is the formation of a structure called the septin ring. So as we can see in this slide, the green object here is yeast and the purple oval shape here is the nucleus of the yeast. Within this yeast cell, here are several subunits to form septin rings, and each of them hold its own function. CDC3, CDC12, CEP7 will form a stable core that help with cell division. Crucial for cell division. Unlike the other subunits, CDC10 is dynamic, meaning it moves between the septin ring and the cytoplasm. It will form a shuttle between the septin ring and the cytoplasm. This is crucial as it helps prepare the cell for nuclear division. Septin proteins CDC3, CDC12 and CEP7 will assemble to create this ring, which serves as a scaffold and plays a crucial role in organizing the cell's internal structures. Septins will form a ring-like structure at the site where a new hyphal segment will grow. This ring organizes proteins necessary for further cell division and growth. In hyphal cells, the nucleus, which contains the cell's genetic material, divides in a unique way. The nucleus moves out of the parent cell and into a new compartment in the developing hypha. After the division, one part of the nucleus goes back to the parent cell while the other part stays in the new hyphal segment. This process ensures that each new hyphal segment has the necessary genetic material to function properly. Let's move on to the next step, which is cytokinesis. In hyphal cells, cytokinesis does not lead to cell separation as in yeast cells. The septin ring splits into two rings and both are maintained after cytokinesis, unlike in yeast where septin rings are disassembled. Septin rings play a role in preventing cell separation post-cytokinesis in hyphae. Special protein, which is MOB2 and CBK1, ensure that the division happens smoothly without completely cutting of the newly formed cell segment. Hyphae don't separate because of different protein. CDC14 is not present. Basically, this protein function is to make the cell divided. The elongation process focuses on the polarization of the actin cytoskeleton, as it plays an important role in hyphal morphogenesis of C. albicans, regardless of cell type. Actin cytoskeleton maintains the directional growth of the hyphae by directing the vesicular flow for the tip expansion. The actin cytoskeleton is made up of two main components, which are actin cables that comprised of long bundles of actin filaments and actin patches located at the tip of the hyphae membrane. Both components are crucial for carrying cell wall materials and enzymes to the growing tip. Polarized hyphal growth are essential to invade host tissues and increase the pathogenicity of C. albicans. This polarized hyphal growth is driven by a Spitzenkorper, a vesicle-rich structure located at the tip, and the function of Spitzenkorper is coordinated by SPA2, BNI1 and BUD6. The first one is vesicle transport, vesicles that originate from the Golgi containing essential proteins for the hyphal growth such as SEC4, SEC2, and MLC1 will travel along the actin cables via the MYO2. Those proteins will accumulate at the Spitzenkorper. The next step is exocytosis, where the materials involved will be delivered to the cell membrane for expansion. The actin cables nucleated by the polarisome complex, specifically the BNI1, will load the accumulated secretory vesicles at the Spitzenkorper onto the plasma membrane by anchoring/docking with the exocyst. One of the exocyst subunits, SEC3, ensure that the secretion of vesicles happens mostly at the tip for optimum membrane material supplied for the development of new cell walls. The polarized growth of Candida albicans also involves endocytosis. Endocytic recycling will take in extra membrane and proteins from other parts of the cell and bring them to the hyphal tip. The actin patches will form the endocytosis site, which they will gather at the tip to maintain the directional growth. In yeast cells, the actin patches will spread out as the bud grows, which causes the entire bud to participate in endocytosis. Meanwhile, in the hyphal cell, the actin patches stay at the tip so that endocytosis only focuses at the tip. Therefore, all the materials will only be delivered at the growing end. There are two regulators that are largely involved in the regulation part, which are RSR1 and CDC1. RSR1 function is to control the amount of distribution of CDC42 and vesicles at the hyphal tip. Meanwhile, for CDC42 is to control the direction of hyphae and affects how hyphal cells grow. So how do these two regulators function? BUD2 will deactivate RSR1 to keep the growth process at the hyphal tip in balance and ensure that the cell grows in the right direction. If RSR1 is overly active, it can lead to misdirection of hyphal growth. CDC24 will activate CDC42 as the active form of CDC42 will help with the hyphal growth. If there is a low amount of activated CDC42, the cells will appear large and round. In conclusion, the pathogenicity of Candida albicans depends on its ability to transition from a yeast form to a hyphal growth form. This transition is influenced by environmental factors such as host characteristics and signaling pathways like the CAMP-PKA pathway. The mechanism involves several stages: initiation, the formation of the septin ring, nuclear division, and cytokinesis. 4. Elongation, hyphal morphogenesis requires the polarization of the actin cytoskeleton.